brats. Brats is especially good at it.
Oh yeah, I'm the best at it.
Get off your phone.
Are we gone? Is it
We're
time?
going.
Welcome everybody. This is episode 149 of the Herpeticulture Podcast. I am Jacob Rotz with Longleaf Reptilia.
Justin Smith, Palmetto Exotics.
And today we are joined by the
Doctor.
one and great Dr. Ben Morrill.
Morrill.
How you doing, Ben?
Good. Thanks for having me on.
Yeah, man, for sure.
So if you listen to the show for, I guess, since the first year, we had been on back in December of 2018. That was episode 21.
So you were one of our first guests, because I think we did 5 to 10 episodes with no guest. Then we started getting guests on after that.
Yeah,
something like that. So one of the OGs.
So
as the kids would say.
Obviously, a lot has changed since then. Both on our end, and we figured it would be a great opportunity to catch up and see what's new, because at the time when we did the first episode, you were still hadn't figured out doing the genetic testing for boas and pythons and stuff, and I know some things have changed since and updated. Let's see. So you were like one of our first 10 guests, I think.
Yeah,
we ever had. I'm looking at it right now.
Excuse me.
So yeah, I mean, I guess before sort of we get into what's new, but a sort of a brief for people that didn't catch that episode or don't know what Rare Genetics Inc. is. What is it? What is it all about?
Yeah, so I, like many people listening, I'm sure, have always been chasing snakes and lizards, and just love that ever since as far back as I can remember. And then through high school and college, I had some opportunities to do some research in herp species, and then as a PhD student, I ended up doing a dissertation on the quantitative genetic traits, reproductive traits in ball pythons. So I was able to work with a bunch of data from Dan and Colette Sutherland from the Snake Keeper, TSK, and do my dissertation on ball python reproductive genetics. And when I graduated, ended up getting a job and moving across the country. And once I finally kind of settled into a new job and being in a new place with kids and everything, took a little bit. But once that all happened, it was actually about the same time that the complete carpet python came out. And Justin and Nick were off in Australia doing awesome things. And I was here trying to get my head back together after the move and everything, and ended up going to Tinley. And at Tinley, that was kind of the first book release, big book release in the US was at Tinley. And so I met up with Sean Christian. He's the person that ran the Morrill Python Forum. He was the second person that owned it, and he's the one that ran it for a while. And so we had talked on there quite a bit, and I talked with him, and we had this idea to be able to start some genetic testing in reptile species. And the thing that we wanted to do the most was get a sex determination test for green tree pythons.
Mm-hmm.
And so we kind of started working from there with different ideas. I did lots of reading and thinking and trying and ordering primers and sequencing and doing different things. And we ended up doing a large sequencing project using RadSeq to be able to narrow down where the actual sex determining sequence is in the green tree python and ball python. We were looking at both just to kind of get an idea for pythons in general. And we went from the 65 million base pairs that's on the sex chromosome in pythons down to about 1 million. And so we made progress, but
yeah, that's a huge jump, though. Yeah. Like, it sounds like a lot, like a million, but when you think it went from 65.
Yes, 1 is much better than 65. Yeah. So it's been difficult since during that time. At the beginning of all that, it was kind of assumed that all snakes were ZW sex chromosomes. But with the ball python mutations, the banana and the coral glow, and people kind of thinking through it, it became pretty obvious that they must be XY, which just means that the species that has two different sex chromosomes is the male in XY. So like in humans, male has an X and a Y, whereas in birds, the sex that has two different sex chromosomes is the female. The female is ZW, where the male is ZZ. And so that makes a difference with, when we're trying to figure out the sex determination test. And so that was kind of a big thing that changed. We realized Warren Booth was one of the authors on that paper, and they published that. And so our RADseq data backed up what he said. Some of the same scaffolds in the Burmese python genome on that sex chromosome are ones that we pulled up as being important. But anyway, it's been difficult to nail down the exact sequence, and therefore difficult to design a test. So in the meantime, I figured out a way to sequence parts of the sex chromosome and be able to follow through the lineage. So if I had the sire's shed and the grandsire's shed, and then all the offspring, I'd be able to follow through markers that are on the grandsire's shed, the sire's shed, and then any babies that have that same marker I know are males, and then any babies that don't have that marker I would know are females. And so it was a little more difficult way to do it, but some green tree python breeders were interested in that. Even though it costs a little bit more and took a little bit more time, it was worthwhile to them. So I've been able to do some sex determination in green tree pythons, but it's been difficult. But now the most recent stuff, and during that time I started doing colubrid sex determination and venomous snakes, and that works great. That's really easy,
much more straightforward.
Yeah, so they do have ZW sex chromosomes, and the Z and the W are way different. In pythons and boas, the X and Y are very similar in size, not like humans. And so that very similar X and Y, it's much more difficult to design a test because they're so similar. There's no differences. It's like trying to tell the difference between really light pink and really, really light pink. Instead of the difference between red and green.
Right.
And so it's been difficult, but yeah, the colubrids and venomous snakes, it's worked great. I, after a couple of, probably about a year and a half. So I've been doing the colubrid stuff for about 3 years, and about a year and a half into that, I was able to design a new, it's a very, very awesome, broad, primer set that does a great job across all these colubrids, and then to also be able to do venomous. And I had a problem for a little while with tricolor hogs and with dry marcon, with indigos, but now I have it tweaked and set up so that it does all the venomous and all of the colubrids that I've tested so far. It works great, so it's really, really nice. And so we have lots of samples come in. We have samples come in on all the time, and that test is going great. But the most recent thing that's exciting, which I've posted a little bit, anyone that's looked at our social media, we found out there's a ball python keeper. He's also part of the Hudson Alpha Institute, Biotechnology Institute, and they're a nonprofit that does a lot of genome sequencing. And they also have a new piece of software called Kufu that is specifically designed to be able to pull out small differences, genetic differences, all the way down to like a single base change between pools of samples. So if we have 50 males and 50 females of a species, the software is really good at pulling that out and being able to give us the sex determining sequence. And then also for morphs, so for ball pythons, we've already started collecting samples and sent some in, and we're working on being able to sequence and pull out and map where the specific mutations are that cause these color and pattern changes in reptiles. And we're starting with ball pythons, but we'll quickly go from that to other species as well, is the plan.
And what's the benefit of being able to find out the location of that
then we can design a test. So right now there's a group of researchers in Canada and then also at Eastern Michigan University that have done some of this work. And from that, I now have a test that I can do. So someone can send me a shed from a posset pied ball python, and I can run that and tell them whether it's het pied or not het pied.
Oh, okay, gotcha.
So then we'd
Oh,
eventually be able to do that
okay.
for all of the different
Yeah.
mutations, sunset, desert ghost, clown, any possets that you have, you can send a shed in and we'll be able to tell you which genes it actually has. And things
That
like
would
yellow
be,
belly, like, you know, if you
yeah.
have a 5 gene animal, you don't know whether it has yellow belly or not. Would
Oh
eventually we would
man.
have a test. You just send it in. What the goal in the long run is to have 30 or 40 of the most common genes, the most common
Mhm.
mutations in ball pythons, and to be able to have it be, you know, fairly affordable. Don't know for sure what that price would be, but you'd essentially pay for a panel. And so we would let you know what that panel of 30 mutations are, 30 genes are, and you pay that amount, send the shed in, and then we'll just tell you of those 30 whether it's het for it or homozygous for it. So if you don't know if it's an OD or a super OD, we'll be able to tell you which it is.
That's
That's
a
awesome.
serious game changer for
Mhm.
the hobby, man. That
Yeah.
I didn't think that would even be remotely possible, but
Yep.
that's perfect, especially for especially if you look at the ball python community and the corn snake community now, there's so many morphs out there, like I have a pair of corns that are het for like 4 or 5 different things,
Yep.
so whenever I produce them, I'm not going to know what the heck I'm looking at, between all these morphs and stuff. So having that outlet to be able to tell you exactly what your snake has, that's amazing in and of itself. But the fact you'll be able to determine a possible het, that's even more, incredible.
Yeah.
That's amazing.
Yeah, we're very excited. It's been fun. There's a lot of people interested and you know, willing to donate money. We have a GoFundMe going now, and if you guys are willing to post that link, that'd be great. But anyone that wants to help
Yeah.
donate some money to that GoFundMe, that's going to go directly to the sequencing costs. And like I said, the first few that we're going to be doing is going to be ball python focused. But if you contribute to that GoFundMe, let me know if it's, you know, you're mostly interested in sex determination and in pythons, or if you're most interested in Western hognose or whatever. We'll definitely be paying attention to those donations and the notes that come with them. And, you know, wherever we see the most interest, that's the next species we'll go to.
Nice.
Well, what's the probability? So, with some chondros and carpets, cancer is kind of a big thing. Is there any hopes that eventually you could have like something that basically gives you just a rundown of sex, whatever genes it might have going on morph wise, and then potential, uh, heritable, health issues?
That's certainly possible. We do it in humans. There are specific tests for
Hmm.
you know, specific types of cancer, and we know in humans which ones are most likely to be heritable and things like that. So yeah, certainly if the work gets done and we know what sequences make an individual more likely to get cancer, you know, in snakes or any of the species, that certainly could be developed, that we could have a test that would tell you that.
Hmm.
That's wow. So it kind of sounds like the sky's the limit with where you guys are going with this, man. That's truly incredible. Everything you've got going on in the works.
Yeah, it's a cool time with, you know, the crowdfunding that's available now, and the sequencing costs are coming down quite a bit.
Good.
In 2000, I was a undergrad, and I remember being in biochemistry class, and they were talking about the Human Genome Sequencing Project, and it was taking years and costing millions of dollars. And now there's an article just this week in Stanford. They just did the fastest whole human genome sequencing to be able to diagnose somebody, and they did it in, it was like 8.5 hours. They
Wow.
Huh.
sequenced the entire human genome, and were able to give a genetic diagnosis for a patient in, you know, less than 10 hours. That's pretty
Wow.
crazy.
Yeah,
And
that's
the
incredible.
costs have come down, so you can pretty easily get a decent whole genome sequence done for $1,000, and in the next few years they think they'll get that down to $100.
Wow.
Hmm.
So it's a completely different ball game than millions of dollars in several years.
Right. Yeah.
Hmm.
That's awesome.
So as far as green trees, though, I want to say Bill Stiegel maybe had posted some pictures of some sheds that he had sent you. I mean, has anything changed with the sex determination as far as those go? Like
Right
if
now,
you
no, it's
okay.
still a little more of a pain. Like for the colubrids
Yeah.
and venomous, that is a very simple test. So that's $16 a test. For the green trees, where I have to do the sequencing and I have to compare
Mhm.
multiple generations, that's $50. So 50. So it's more expensive, more of a difficulty. And whoever's submitting the sheds, they have to have, not only the offspring they want to sex, but the sire, and either the grandsire or a known male offspring from that same sire. I just have to have a way to track the markers
Right.
on that Y chromosome.
Okay.
Makes sense.
So yeah, so that's still the same, but I will have and Ian Bassell and some other people have given me sheds. I'm going to have some green tree python sheds in one of these first big runs with Hudson Alpha when that happens. And so I'm hopeful in the next 2 to 3 months we'll have some python sex determination tests going.
Awesome. Our buddy Jason Keller actually talking about venomous. He's big into heloderma, and his question was this was added to the outline sort of later in the day, but figuring out the gender of heloderma or lizards in general.
Yeah, so heloderma are definitely on my list. Varanids, also blue tongue skinks. All of those are ones that I would like to do. I don't know if we'll be able to get to them this year, but with the decreasing sequencing costs, and then Hudson Alpha helping with their software, the Kufu, it'll be much more likely and definitely much faster.
So with something like, lizards or, or basically anything, lizard wise. Yeah. And again, since y'all aren't doing it, I don't expect you to know the answer for a fact, but it seems like it would be much more difficult because lizards shed in pieces, type deal, or they rip it off with their mouths. I know a
Just
lot of
have more
geckos.
samples.
Yeah, I know. Well, I know a
Just
lot of
spread
geckos.
everywhere.
Yeah, so I'm wondering, like, would that be a situation where you just get as many pieces as you can and send them off, or is it like, no, it has to be like a ventral piece? You know what I mean? Like a spot from a certain part of the body, or does it matter, or can it just be
Yeah.
one single strand of shed? Or
So what I have on our website as far as like instructions when people want to send in sheds, the kind of rule of thumb I give is to try to give us a piece about the size of a quarter.
Mhm.
From that big a piece of shed, I can do probably 30 to 40 extractions.
Wow.
So the size of a quarter is a lot, and
Mhm.
it doesn't matter if it's in multiple pieces. Really, I'm using a piece probably about the size of the top of a number 2 pencil eraser.
Mhm.
So even
Wow.
smaller than that most of the time. So yeah, even if you have just small pieces, if you want
Mhm.
to find it on the website, if you go to how to submit, yeah, right there. So how to submit samples, and I've got an FAQ in this section as well that kind of gives you some answers to frequently asked questions. But yeah, the size of a quarter is plenty. So I have lots of people that will send me, the full shed from adult, large colubrids, and I'm only using a little tiny piece.
Yeah.
Dang. Well, that's good to know. That's awesome. Yeah, because I know, like with geckos, I can only imagine it being a little bit more difficult because I know leopard geckos and I believe cresties eat their sheds.
Mm-hmm.
Normally, like they'll rip them off and just eat them. So I know that
Yep.
you have to kind of catch it at the right time.
Yeah.
But nice.
Yeah, that definitely could be more complicated. I haven't had to try to take sheds from lizards. I know sometimes you'll see lizards, they'll have, little pieces of skin, kind of a stuck shed going on.
Right. Right.
So that would be easy to just pluck off a little bit, in the case like that. But even if you're finding some, if it's in a dry area of the cage, the thing that is the biggest problem is when you have wet conditions. If the shed is exposed to moisture for a prolonged period, then it degrades the DNA.
Oh, I
But as
got you.
long as they're in a dry section of the cage, like in the bedding or whatever, and it's dry, then most likely, and it came from that animal. If there's two that had been in there breeding or whatever, and you don't know which one it's from, obviously you can't use that. But
Yeah. Well, we know plenty of people with heloderma that can probably send in some sheds. I'm reading here. Y'all are looking for samples of known gender to
Yes.
contribute to the cause. My Ackies, I'm fairly certain are a pair, but if I ever get a solid idea of what the gender is on those, I'll send in something for those as well.
Sounds good.
No,
Yeah,
we know
like I
plenty of
said,
people.
right now. I wouldn't. I mean, people certainly can send them in, especially if they're known gender. But like I said, I don't know for sure when I'll be able to focus on that.
Mhm.
It would be nice if it was kind of second half of this year if I could start working with some of those, but I just don't know how all of the morph stuff is going to go and how long that's going to take.
Right.
There's just so much stuff to do and no time to do it.
Yep.
Yeah, the reptile hobby is there's so many different aspects to this hobby, and everybody's gonna want their aspect looked at, because this is such a niche, thing that you're doing here, and I know everybody's gonna want to be, a part of it somehow. So the lizard guys are gonna be like, focus more on lizards, and then the morph guys are like, no, keep going with the morphs, and so I can only imagine
But
the
I
trying
am
to decide.
working on the first lizard project right now with panther chameleons.
Okay.
And so some of the panther chameleon keepers got together and raised money to be able to develop a test to tell the locality
Oh,
for
wow.
panther chameleons. So I didn't know very much about panther chameleons before that, but ends up that there's this really cool study. There's some researchers that went to Madagascar and they collected across the whole range of panther chameleons. They collected, I think it was like 330 blood draws from wild chameleons, and they took pictures of them and have locality data. So they have not only the coloration in a picture, but they also have locality data, and they got DNA. And then they sequenced and published all of that information. So that's a huge step to being able to develop a test to tell a locality or some kind of purity or something like
Mhm.
that, because we have over 300 known wild um, samples that I have sequence for, and I can align any panther chameleon samples coming in. I can align them to those and then put it in the groups that this publication they came up with from the DNA sequence. So that is one lizard project that's going and looks good. They did get the money together and they're collecting sheds, and I've been doing a literature review and coming up with the game plan. And I think within the next month they'll probably send sheds in and we'll get the first testing done. And it will be interesting to see if the captive chameleons, how many of them look like they, have similar
Yeah.
sequences as the wild
Right.
play, the wild localities, or if that looks like there's a lot of mixing that's happened. And so it'll be interesting. And the females are pretty brown regardless of where they're from, and so it's difficult. You have to trust the exporters that they're telling you the truth. And so it'll
Right.
be really interesting to see how that all shakes out.
So with the locality thing in mind, do you think that's something you could work towards with carpet pythons as well? Because that's a huge
Bear
thing
tracks.
in the carpet world of,
Oh
is
yeah.
my jungle pure? Is my coastal pure? It's a little easier with, poplins because we can still get them imported, but even those have been crossed and all types of things.
Yeah.
So
Or
I
even,
think that alone in the carpet community would be
yeah.
huge, being able to, because there's, I think of like a lot of the diamond lines, specifically,
Hmm.
there's always talk of, some of these diamond lines having jungle blood in them. There's people fighting about it, horrendously. I'm not into either one, so it doesn't bother me at all. But I
Yeah.
know that could be very, very helpful for a lot of people in the carpet community.
Yes. Yeah, so I haven't looked at the literature specifically. I would have to look into that and see. We have to have DNA information
Mhm.
from wild caught.
Yeah.
You
Kind of
Gotcha.
like
know,
that
you
baseline
have to have,
information.
yeah, you have to have that base. That's why they're so lucky with the panther chameleons that someone
Mhm.
went out and took that many. I mean,
Right.
just doing 330 blood draws from wild lizards, I'm imagine that took a lot of time, especially
Jeez.
because they're from all over that, all over Madagascar.
Yeah.
So yeah, I would just have to look and see, and then what access we would have to like zoo samples or other people that would have ones that have locality data. But as long as we can get kind of that footprint of what they look like in the wild, and we know what their locations are, then yeah, we could definitely develop a test. And people that post what is my carpet, we could give them kind of like when people say, send in a sample from their new puppy they got, and they find out what percent, this breed and
Exactly.
that breed.
Yeah.
That's definitely doable. We just have to have good wild caught samples to compare to.
Well, if anybody, I know we have some listeners in Australia, so if any of y'all field herpers out there got any ideas, get with Dr. Ben over here. Can get something going with that in the future. That would be cool, man.
That would
I
be
think
great.
it would be really helpful with the bamboo rats.
It'd be really helpful with a lot of stuff,
Just
man.
as well,
Because you have so many people that are into the locality specific stuff, like myself, and being able to determine the purity of that is huge.
yeah, but the bamboo rats in particular, those get crisscrossed a lot, and they're very hard to tell apart from one another, especially if you have hybrids.
Right.
Because they were just talking about that on Colubrid and Colubroid Radio with Rob.
Mm-hmm.
And that's always been one of the reasons like I haven't gotten into bamboo rats, is because like if I get something, I want it to be one thing. I don't want it to be crossed. And
Mm-hmm.
there's just a lot of crosses out there
Yeah.
in that group.
Yep, it's all possible, and it's getting cheaper and more doable. So it's at a point now where, like with the Chameleon project specifically, where I knew we already had some DNA data, with $5,000 that they saved up, we're able to do the literature review, get sample or get primers ordered, and be able to test the first 25 samples, and kind of get on our feet. And maybe we'll find out we need to do some more tweaking to that test. I don't expect it to be perfect right off the bat, but same with any of these other projects, especially if there's some good data that's already, and with a lot of the species there are publications from researchers that have done some sequencing. And so there may be some other species that would be pretty fast and easy to do that. I'm also with Morrill boas and Madagascar ground boas. There's another project that keepers had funded to be able to tell the difference between those two.
Nice.
So that's another one that I'm already have gotten sheds and have been working on. I found some pretty cool. There's some that they feel like are pure, and then I have some sequences from published data from scientists to compare to, and they have some that they thought were crosses, and I can see a clear signal in the DNA that looks like that's what it's telling me, is it's a cross. But I need some more pure Morrill samples for that, but that's coming in soon, and so that'll be interesting too. We may have a test to be able to tell those two apart.
Man, that's awesome.
In the event that you somehow end up with some free time to do other things, we had Rob Stone on Snakes and Stogies. Was it last week? No, the week before last.
Yeah.
And we were talking about Kylobothris, Puerto Rican boas in particular, and he was talking about how there was a study done on ones in the U.S. hobby, because we don't have access to the wild populations. And he was saying that the U.S. population, everyone thought that it would be diverse, like genetic diversity wise, it was very, very watered down, very shrunken. There wouldn't be a ton of genetic variation between them. But he was saying there was actually just as much diversity than they've seen in the wild. Is there a way that, will there ever be a time that you can do something similar with some of these species like rock pythons, the other stuff like Jamaican boas too? They haven't done those. Just stuff that like we're cut off from getting new stuff in, but we can tell how far down that hole we're getting.
Yeah. Yep. There's pretty well set out genetic tests. Like there's something called an inbreeding coefficient, and your percent heterozygosity for specific, sequences, things like that, that you can look at pretty easily, and be able, as long as you can get those wild caught samples to compare to, then yeah, I really like that. And I like the idea of especially commonly kept species, like ball pythons, corn snakes, king snakes. I think it would be great for breeders to be able to have some kind of inbreeding coefficient available, like on their website. Say, hey, we've got our breeders tested, this is our inbreeding coefficient. And that would be a way for people that have worked to make sure that they're outbreeding and keeping their animals, having hybrid vigor, that they would get, some way to advertise that to their customers.
Yeah, because that could definitely be, a concern to, some people. If you want to get a pair from somebody, it would be nice to know, like, okay, these are genetically outcrossed enough to get a pair and be okay to breed that pair together, from the same clutch, versus this is pretty close to home, and so that's amazing.
Yeah, and that's something indigo breeders have been doing for a while. They actually worked with another genetic testing business and got a test up and running. It sounds like it's a really long lag. That's something I would also like to help as an identity test for indigos, but they found that to be really important to do that identity test. And then when they go to do their breedings, they make sure they have animals that are as genetically diverse as possible to avoid breeding, their offspring having different inbreeding problems.
Right.
As far as that test for the inbreeding, how does that work? Do you have to have sheds from the parents to, like, how do you know how much something is inbred in that
So
test?
what they would usually do is they're going to test all the adults they have, and then they would just breed the ones together that are the most different. But the other way you can look at it is there could be some sequence we would focus on, and you would sequence that in an individual, or multiple locations, and then you would say, okay, what's the percent heterozygosity? And so a really common test, I think this is how the Indigo test worked. They use microsatellites. So they're just these highly variable genetic markers throughout the genome. I think they had 15 or 17 of them. And so then you would get a percent heterozygosity. And the more heterozygous they are, that means the more outbred they are.
Okay. Now, having seen all the samples and stuff that you've done, especially with ball pythons, is it safe to say that a large percentage of the morphs and stuff that we have in the hobby are pretty deep in that inbred sort of category?
I haven't really looked at the inbreeding with ball pythons. I do know, back in the day for my dissertation, I did some sequencing in ball pythons and carpet pythons. And specifically, I was looking to see if I could show two snakes coming out of the same egg, whether they were monozygotic twins or whether they were identical twins or fraternal twins. And from that, I was able to be the first to have some non-avian reptile and, well, I guess, turtles they've shown twinning and identical twinning. But this is the first time outside of birds and turtles where we had some monozygotic twinning. So I was able to show they were identical twins. But from that, I did have some samples from granite carpet pythons, and those definitely got heavily inbred.
Yeah.
That was the exact example I was going to use for inbreeding, because I feel like that is like the perfect example of too much inbreeding. Like I used to keep, I had a het
You did
granite
have
and a
some
granite.
granites.
I had a visual and I had a het, and
Hopefully your het was the female.
no, actually the het was a male and the visual was a female. Yeah, I don't have
Dang
either of
it.
them anymore, but they were weird. And nobody like believes me, but I'm telling you, they were weird snakes. Like their immune systems didn't seem as strong.
Get your tinfoil hats on.
They were just odd. They were odd, you know, and I didn't say much about it, you know, but they just seemed more fragile. I don't know, you know, but like I've seen some messed up granites that act like jaguars, you know, and it's I don't know. I'm not granites are a perfect example of way too much inbreeding. They got some really weird
but it would be interesting now that it's been, because that was in 2011, it would be interesting to get some samples, especially from breeders that have specifically outbred and made granites, and see if they've been able to improve that. And my sample size was really small. There's just the few that I looked at were very homozygous, very heavily inbred.
Yeah, I know a lot of people have been working on the Granigen because it's a beautiful mutation. I love it, but you know but I know a lot of people have been working on outcrossing that, and you have some very healthy granites. Now, nobody attacked me for saying I think granites are kind of weird. That was just my personal experience, but yeah, no.
Well, I only had one het female and a het male, and she only got pregnant one time, and she got egg bound, and I think that she ended up getting some kind of infection. I was able to get the eggs out safely, but I think in the process she must have had some kind of a tear, but within a few days she died. So
that's unfortunate.
Yeah, that sucks.
Yeah.
So go ahead.
I asked about the ball pythons just because I'm always wondering, I'm not a ball python guy. I will never really claim to be. I have no clue what I'm looking at half the time with those things. But with the amount of mutations and combinations and stuff now, I've always, every time I see some at shows and stuff, I just wonder, how much of that is a result of that just folding the genes with each other, back and forth,
breeding them back, because that's what everybody does, for these hets and stuff. And it's because I can only imagine there's so much more of it with these recessive mutations, because that's what people do. They get hets and they breed them right back to the parents or siblings, and to try and get more, and then you just produce more and more and more from siblings back to parents, and then
do with the Henry Bairds.
Yeah, but see, that's like a first or second generation thing, like if you do it,
I'm not worried about.
Yeah, if you do it for a couple generations and outcross, you probably be fine. But like if you just breed siblings to siblings and back to parents for 10 years straight, like, and then you're curious why these things are coming out all screwed up, it's like, I don't know what to tell you, dude. Like if you go have a baby with your cousin, it's not gonna work out.
It's a little different,
like I know it's different, but still
different.
I know it's a tad different. I realize that, but all I'm getting at is at a point you're gonna hit a wall. There's got to
learn where babies come from, Jake.
Oh my God.
Well, I think specifically with ball pythons, the vast amount of incomplete dominant traits probably saved a lot of that, because people want to get 3 or 4 or 5 incomplete dominant traits in with their recessives, and so they're forced to outbreed. Whereas if there were only recessive mutations, I think it would be much worse.
Yeah, and I think that's why the granites got so bad, because people were trying to produce granites so much, but that was one of the only morphs in popwin carpets. So they just kept breeding them back, and but it's looking a lot better for granites nowadays, though.
That's good.
Crossing them into a lot of other stuff, and it's there's some good looking animals out there for sure.
So what about the potential for doing like the way I described it in our outline is the potential for like an open source sort of genetic mapping? Like I mentioned chondros specifically, but since we're not quite there yet, we can switch it to something that works a little better. Like when you get enough samples in over a course of time, being able to look at that and show, like a just a giant map that all these animals came from, these other, I can't explain it in a way that makes any sense, but just a giant like web that just shows where a lot of these base animals that a lot of these animals came from. Is that something that you would be able to do?
Yeah, that's
I don't know how much data that collects and takes up, but
Yep, that's definitely doable. because for my dissertation, the department I was in was an animal science department, and so I'm familiar with the projects people have done in the food animal and sheep and things like that, and they're able to come up with these that they're able. There's a lot more money in those species to be able to do these genetic tests. But yeah, they're able to track and figure out like exactly where, like if there's a mutation that's helpful or a mutation that's hurtful, they're able to track and figure out where that came from, and they know which sires to not use anymore, or which ones you want to make sure you're using because that's a beneficial trait. But yeah, you're able to do that kind of work. That's definitely doable, and I think green trees would be a great species to do it with, because people that keep green
especially the
trees,
designer stuff.
yeah, they seem to do they seem to have more of a focus on the lineage and the history, whereas if you buy a ball python, the chance of getting information about the specific sire and dam or their ancestors before them is close to zero.
Yeah.
I figured designer chondros would be perfect for it, because there's a lot of the sort of the early designer animals. They were the founders for so much of what we have now, and so it'd be cool to be able to look at a map and be like, I have this animal from this pairing that had a John Irby animal in it, like one that I have from Luke, and being able to trace that one back to the grandparents, and then finding out someone else across the country has something that was also from the original base pairings and stuff like that. It seems to me like there's enough of those to make it sort of work, but it's not so massive that you're never going to find any sort of links,
Yeah. Yep. Yeah. With that Indigo project, they were able to find some markers that went along with lineages, and so there's specific markers they'd see, like, oh, I know that came from that sire 5 generations back. So yeah, that you just have to start getting some of this testing done and get kind of a database started to get put together for that species or that group or whatever, and you'll be able to start telling some of those things, which is pretty fun.
Nice.
That is one of the things I love about chondros, is just that there's no other group in herpetoculture that I think takes as meticulous care and notes of lineage as the chondro people do.
Yeah, that's great. I love seeing that.
For sure. Well, with all this going on, and all the different things you've got going with rare genetics, do you do any captive keeping yourself?
Yes. Yeah. So I listened to one of your recent episodes. I saw it had king snakes in the title. It was a professor from West Virginia.
Dr. the Crawdaddy.
Yeah, that was last week.
Yeah,
Yeah.
I never thought about keeping Nerodia, and I've definitely never heard anyone talk about keeping Nerodia. That was super interesting.
Man,
Nice.
they're making a comeback.
Yeah, I mean, I've caught them now that I live here in Virginia. I've seen them, and I'm one of those people that dives into the water catching them.
Yeah, man.
Because I try to catch pretty much it. The one snake that I don't go after are garter snakes, just because I've gotten musked by lots of garter snakes since I was like 5 years old. So I am not as interested and excited to see garters anymore, just because I did lots of times growing up. But the Nerodia are new for me. I didn't have those in Utah, so now that I'm in Virginia, I chase them down. But yeah, that was pretty interesting hearing that. But the colubrids that I keep, I have Hondurans now. I've had gray bands in the past, and I've had speckled kings. I think that's probably it for colubrids for me. But yeah, I've got like 10 or 12 juvenile to adult. I got my first, I guess we had 3 or 4 clutches of Hondurans this year.
Nice.
So those are pretty fun. I just keep them in the bottom of the rack where I have the ball pythons. So I mostly have ball pythons now.
Okay.
I've kept Angolans, kept inbred Angolans and blackheads and womas. I've obviously had many of the carpet pythons. I didn't ever breed any Brettles. Justin produced all of those when we were working together. But yeah, I've kept several different python species. But yeah, right now it's just ball pythons and then the Hondurans. And I can tell you that light cycle is not needed for the Hondurans to go, nor
Yeah.
is much of the temperature difference. I literally just kept them in the bottom like 2 slots of my racks that the ball pythons were in and did nothing different. I still fed them, here and there through the cooler times. And they're in a room with no windows, and so there's only light when I'm in there. Other than that, it's dark, and they cycled just fine. So for what that's worth.
Nice. So do you, with having, your own captive animals, do you use them towards any of like your testing and stuff? Or do you have animals that you got in mind specifically for rare genetics?
Not really gotten any specifically for, to be able to have a source. I thankfully have been doing this for so long and gone to a few carpet fests and, lots of reptile shows. Whenever I need sheds from various species, I know who to talk to.
Yeah, for sure.
So I can get sheds pretty easy. But I definitely have like all of the colubrids that I hatched out this year. I kind of popped them a little bit. I popped tons of pythons, but very few colubrids. And so I did a genetic test on all those just to make sure. And it seems like I can pop okay with colubrids too, but I'll probably keep running the genetic test because it's so easy for me to do.
Yeah.
But now that I have this het pied and het lavender albino test in ball pythons, I went through and got samples from all of my, well, not all, but about now it's probably 30 or 40 of my animals that are either pied or het pied, and ran all of those to make sure that test is working properly. And I had some pos hets I was able to prove out, and I know I've got some animals that are worth about 2 or 3 times more now that I know they are het pied.
Yeah, exactly.
Yeah, Terry Burwell used the testing for the cyania he got from me.
Oh,
which
okay.
I loved because they then confirmed that I actually did pop correctly and that I
you
did
got a
actually
pair.
get a pair. So I was like, yeah.
Yeah,
those in particular, man. Like I talked to guys over in Europe after I hatched them. I'm like, how do y'all sex these things? He's like, yeah, we just pop them. I'm like, dude, if I pop this, I'm gonna break this damn thing.
That's
like,
how
it's
I
so
feel about
tiny.
like the little baby corns.
And he
man.
was like, no,
like,
man, it's cool. He's like, they handle it fine. I was like, what?
That's
sure
that's
enough.
how I feel about like Chris told me like with like Paint Chat with all his baby corns,
Yeah.
he just pops them. And I'm like, dude, I wouldn't pop this thing like after it's had 10 meals and you sent it to me. Like this is tiny.
I forced myself to start doing it and like trying to get better at it within the last year or two. And like I said, it seems like between that and sort of the the visual shed deal that Stu Tennyson has talked about with the older animals, but I've gotten better at it, and it was because I was like, all right, I got to learn how to do this. I've tried probing in the past. I don't like really don't like doing it. It makes me nervous. But the popping thing, especially if they're like fresh out the egg, I figured out the right amount of pressure and that kind of thing.
Yeah,
so
and you have to
put
a weird amount of pressure into it, like an uncomfy amount of squeezing. I got good at it with carpets.
It's all about the roll.
Yeah, it's all about the roll you get. I did it with both carpets, but I did the same thing. It was like the day they hatched, while I was separating them, I was popping them while I separated because that's always what I've heard. The day they hatched, it's easier to do it,
especially with carpets, they're less likely to bite you right out of the
egg. So I did all that with all mine, and as far as accuracy, nobody's bred anything I produce, so I can't say that I've been 100% accurate, but I'm pretty sure I only sold males my first clutch, and then this one I haven't sold. No, Billy got a female for my first clutch. But yeah, I'm pretty much only sold males. So
practice makes perfect. You just have to be gentle.
But the males are easy. You know what I mean? Like if you sell somebody a male, it's a male. It's the females that you question.
Yeah.
It's always
false negatives.
You'll get those couple guys every year. They're like, yeah, my entire clutch was female. And then it's like, if I got that,
half.
Yeah, exactly. But there's some people, like I know a guy that was like, he's very good. I've been doing it for years. He's like, dude, this entire clutch was female. He's like, I popped these snakes 3 times over each.
Yeah.
He's like, nothing changed. He's like, they're all females,
I got one ball python clutch of 10 eggs 2 years ago, and all 10 were female. That's the only time I've had that many. And
That's
the
crazy.
same thing. I popped them 3 times. I usually pop with ball pythons right out of the egg after their first shed, and then either when I sell them or when they're, you know, like 6 months old or so. If it's a female, obviously, if it pops male the first time, I only pop it once. But
Yeah.
Right.
I usually pop them 3 times, and I would say 1 or 2 out of 100, I get it wrong on the first pop. I'll think that it's female, but on the second or third pop, it'll pop a peen.
Yeah.
It happens. If you're not careful, you can miss. But
Yeah. And you
but
can also,
here soon we'll have tests for that too. And
yeah.
so it's, you know, $16 for one, but if you're gonna send a bunch in, it's $11 if you're doing 25 or more, or 26 or more. So
Right.
if it's a species you're not sure about or you don't want to get good at it, you have got another option.
Yeah. For sure.
Even just for confirmation.
Yeah.
Yep.
Because that would be my
There's
thing. Is
always
like,
going to be a shred of doubt in my mind.
You could just send the ones in that you didn't get to pop.
No,
The ones
or
you get
just-
to pop, don't worry about it. The ones you didn't get to pop, send them in and make sure they're actually female.
or just send in the ones that you think are females. You know what I mean? You got to
Yep.
clutch of 10, and you have 4 that you think are females. Just verify those 4. You know what I mean?
Yep.
Because you know it's easy to, you know, and like even with baby carpets, man, it was weird. Like popping those, you know, you have to know what you're looking at, because it's not, the males aren't prominent. It's literally 2 red dots that you see. It's not like you have these 2 hemipenes that evert out and it's obvious as all get out. It's literally
not
just
well endowed.
it's just 2 little red dots that come out, and you
Yeah.
gotta know what you're looking at, because you can barely see it between your fingers as it is, you know. But yeah, no, it'll be very, very helpful going forward. I'll do that for females. Heck, I'll send in skins and make sure. I would do that all day.
It's kind of nice to know when they're newborn instead of 3 years later and find out you got it wrong.
Yeah.
Exactly. And I tell you, nothing would make me feel worse than sending somebody a labeled female that was male. You know, that's
Yeah.
happened to several of my buddies. Like, man, I raised this thing for 3 years. I bought it off this
Yeah.
person, and it's a male, you know, like, oh God.
Trust but verify.
Yeah, exactly.
Yep.
You know, and
One of my fun memories was I had a killer Irian from Yasser. It was a super nice male.
oh yeah.
And good night, buddy. My son's going to bed. So this is a super nice male that I got from Yasser years and years ago, probably like 2005 or something like that. It was like so light and bright, lavender and orange, really cool male. And there's someone on the forums that had a female, and so they sent the female to me for a breeding loan. And I came in the next day after I paired them together, and that big old female had a nice giant peen that was trying to get that my male with.
Oh man.
Sorry,
Just
buddy.
a big old male that was trying to force his way onto my male, and so then I
no.
separated them and sent it back.
It's like they were cellmates.
Yep.
Jeez, man. Well, that could have gone much worse if they started. It started
It's
to combat
true.
or something.
It's true.
you
At
know?
least it was love and not fighting.
Yeah, because that male.
He said one way or the other, it's happening. buddy.
We're getting something out of this. Jeez, man. Well, yeah, no, it's so unfortunate. I had a buddy that had a higher end project. I believe it was an exanthic pop one project, and he raised up a female for years and turned out to be male. And it was like, oh man, that's
Yeah.
a real kick in the nuts.
That's a bummer. And
Yeah.
you know, here pretty quick, just less than $20, you can find out. And if you're doing a bunch, closer to $10, you can find out.
How long does it
Nice
take
and
to
easy.
do a single sample? Like you get a shed, start to finish until you have results. What's how long does it usually take you to do those?
The turnaround time?
Not even the turnaround, just like the sample itself, like testing that.
Yeah. So when I get samples in, I'll sit down once I've got a group of at least 20 or so. Most of the time it'll be 20 to 50 that I'll sit down, and I have to carefully with tweezers pull off that little small piece, put it into a little tiny PCR tube, and then I flame sterilize, go to the next one. So it's pretty fast getting the sample in the tube, setting up. And then once I've got those 20 to 50 all in tubes, then I'll move to the PCR, and I have to extract first and then PCR. But anyway, so like if you say like a group of 20 samples, I can usually have the shed prepared and the DNA extracted and the PCR done, and then results from start to finish. If I was doing a group of 20 straight through, which with a full-time job and 4 kids, that rarely happens straight through, but
Right.
probably 4 or 5 hours if I was
Okay.
focused just on that.
Okay.
But there's a lot of fixed costs and fixed, amount of time, whether I do 1 sample or 20.
Right.
It's only going to take me a little more time to do 20 than it would to do 1. And so I pretty much always batch them because it's just not efficient at all to run, less than 10. It's kind of a waste of time.
Yeah, that makes sense.
That makes perfect sense. Yeah.
Have you done anything with the Hondurans? Because I know, if I recall, there's some sort of confusion or consternation about how legit the Hondurans we have in the hobby are. Is there?
Yeah. They talk about them as being hobby Hondurans, which just means you don't know for sure what they are, but we call them Hondurans. I've just done sex determination. That would be a fun test. I've got, as far as king snakes go, actually, yeah, I think they are king snakes. There's one breeder that sent me probably 500 or 600 sheds.
Holy.
Yeah. So it would be a pretty fun project. It just takes time and money.
I want the results on my desk by Tuesday.
We could learn a lot about those king snake species that he sent, and with the information of where he got individuals and from different sources, and to see what the population structure is like in the hobby. I would love to do projects like that. There has to be some way to be able to focus on that time wise, and I can't do it right now, but that's something I would love to do in the future. Another thing I'm interested in doing is figuring out a way to have like a scientific journal, but it's specifically for captive bred reptiles instead of
That'd be
right
cool.
now. Really, if you want to submit to a scientific journal, they're not really captive bred focused. But like if you look in cattle or chickens or whatever, there are journals that are for these animals that are raised in captivity. So it's not something new to science to have journals from captive bred species. It's just nobody's done that with reptiles yet, and I would love to do that. And if we do that, maybe we would be able to get some more studies like that done, because people would know it would get to be published, people would know about it and be able to see that, and people would refer to it. Also be a way to promote your business if you're taking part in those kinds of projects. That might be a way to get people more interested and get some more money to be able to do projects like that. I would love that.
Nice.
How similar is the testing that you do to something? So when I ask this, I have the rhino rats in mind. There's that new species, the Hynenensis. The DNA work that they do on those, is that similar to what, like, testing wise, is it the same, similar to what you do, or is it a different?
Probably. It's probably similar to some of the stuff that I've done. I'm not familiar with that specific, like that literature. But most of the time what's happening is they're looking at a mitochondrial gene or two, and then a couple of nuclear genes, and they're looking to see, and they use, it's really cool. Some of the stuff that I did as an undergrad, actually on some toad species, but they're usually using molecular clock, however many base changes there are between the two groups they're comparing to figure out whether they're different species or different subspecies. They'll see how many base changes there are, and they have other information to kind of put into this software, and it will give you these trees and tell you, the likely amount of time that these two groups have, if they've been separated, if they have been separated, how much time there's been. So yeah, a lot of that is you just have to find these regions of the DNA, specific regions that we have some information about how frequent we would expect there to be mutations. And then that you can use that kind of like carbon dating for rocks and things like, carbon containing things. it's the same thing in the DNA. There are certain parts of the genome and the mitochondrial genome, the nuclear genome, that we can look at, and that's kind of like a clock that we can have an idea how distantly related something is.
And is it common for people to use both mitochondrial and nuclear? Or
in studies like that, that's pretty
Okay.
common that they would have some mitochondrial data.
Because I've heard there's some debate between the, whether one is better than the other, or one indicates some like nuclear being a better indicator over mitochondrial, but I'm not familiar with them enough to really to know either way, but nope, you're muted. We can't hear you. Doc,
Can you hear us? You're muted.
we can't hear you. You're muted.
Nope.
Womp womp.
Well, it says your little microphone by your name has the mute button on it. Oh,
I can't unmute him. You muted yourself. Your mic isn't connected, it says. Nothing.
no. If you look on your little icon down below, there's a little microphone button there.
Hey,
There we go.
I wasn't even moving. I don't know.
Yeah, who knows?
Yeah. It's the internet.
Now I don't remember what we were talking about. Oh yeah, so the mitochondrial DNA. The biggest difference between mitochondria and nuclear is the mitochondria come from the egg, and so there's only a genetic contribution from the mom, whereas
Okay.
the nuclear DNA, there's the sperm and the egg came together, so you have genetic information from the mom and the dad. And so the mitochondrial DNA is less likely to have changes over time because there's no crossing over, there's no segregation. It's all just, copied down from that maternal ancestry.
Okay, that makes sense.
Cool. Yeah.
Oh,
What else we got on this?
I feel like we probably asked you this on the first time we had you on, but what species do you receive the most of?
Definitely the test I run the most is the colubrid sex determination test and corn snakes. I get truckloads of corn snake samples coming in. Thank you to all of you corn snake keepers. And they seem to spread the word and on social media, let people know. And yeah, we get lots of corn snake samples coming in.
What about how often are you getting venomous samples?
I would say compared to colubrids, maybe 1 out of 50, 1 out of 70, something like that.
Wow.
So much less common.
I feel like that what you do is almost most beneficial for venomous keepers.
Yeah, no kidding.
For venomous breeders.
I think so.
I would, man. I don't know how. I mean, you could. I guess there's always tubing, and you can pop doing that. But man, geesh, no way, man. I would.
Yeah, I don't know.
I'd be sending everything your way.
I don't know if it's because there's just a lot less people keeping venomous, or if they're not as worried about what the sex is. I don't know, but yeah, we don't get very many.
And it seems like for everybody that keeps venomous, there's even less that breed venomous.
Yeah,
I feel like there's plenty of people that have
customer base just shrinks the further down you go.
Yeah, there's plenty of people that have their little Atheris or their squams, little rattlesnake, copperhead here and there, but there's not that many people breeding those things. So the need for it, I guess, wouldn't be as high in that regard. So.
Yeah.
But it's still either way, if as that progresses, and people are breeding more venomous in the future, that would be a huge tool.
Yep.
Cool.
It's nice to not have to touch and poke and prod. That's for sure.
Yeah, that's
the less skin to skin contact, the better.
Yep. Not to some people though. I'm sure there's plenty of guys out there that would stick their finger right up a king cobra's butthole. They could. Jesus.
What about crocodilians?
That's interesting.
Are they they're temperature dependent sex determination, aren't they?
I don't know if all of them are, but I know alligators are.
Yeah,
Yeah.
I know most are, but you can still hit like that right in between and try and get like a half and half type ratio type deal.
Yeah, so as far as a sex determination test from DNA, if it's temperature dependent, then there's nothing in the DNA to tell you sex.
Mm-hmm.
Gotcha.
It's all dependent on the environment. And so
Okay.
species that are temperature dependent, we wouldn't have anything. Even bearded dragons, they have a very interesting phenomenon where if you incubate the eggs at a higher temperature, that forces them to be one sex. I think it's male. I can't remember for sure.
Mm-hmm.
At a lower kind of normal temperature, you'll get genetic sex determination. And so there is, sex chromosomes and genetic sex determination. But if you cook the eggs warm enough, they actually just end up being one sex. And so
Hmm.
I still would be very hesitant to do a sex determination test for bearded dragons, even though they do have sex chromosomes. But if you have the environment a certain way, then that test is no good.
And are you aiming for this to at some point be full time?
It'd be fun. I like my full time job. It's a good job. I like who I work with. But yeah, I mean, if there is enough of these projects take off, enough things going on, I could see it become a full time thing. That would be fun too.
Mm-hmm.
Yeah.
And have you worked with Warren on anything?
We've talked about things several times.
Okay.
I've never ended up doing a project with him, but yeah, he certainly is thinking about similar things all
Mm-hmm.
the time. He's more boa minded. I'm more
Yeah.
Python minded, but yeah, we definitely have a lot of similarities. The things that we spend our time thinking about.
Yeah. Man, I wouldn't even want to know all the things us reptile keepers think about on a daily basis. All the crap that goes through our heads 24/7. It's
Brumation's been heavy on my mind lately, man, after all the conversations we've had about it, with Zach and stuff. Not even on the show, but off,
last
in the
week.
messages and stuff. It's
Yeah.
like I'm constantly wondering. I was talking to my buddy Gabe Schuler just yesterday. He sent me a picture. He found a cottonmouth and it was like 38 degrees out.
That's
And
crazy.
I'm like, and I told him, I was like, dude, the more of that kind of stuff I see, the more I'm convinced that the stuff we have here locally truly does not go down for any considerable extended amount of time.
And that's exactly why, like, when last week when Doc said the whole like going about these southeastern colubrids more like we breed carpets,
Mm-hmm.
as soon as he said that, I was like, oh my God, like that actually makes perfect sense. Maybe drop them a little bit more than a carpet needs to at night, but the idea of cooling them down at night and bringing them back up during the day, I mean, that's exactly what we go through, especially, here in Beaufort. There'll be plenty of nights where it'll get down to, 40, low 50 degrees, but then the next day it's 80, or during the day it's 80 or high 70s, and it's a great time to go find snakes, so it's.
Well, even then, I mean, given the last couple Decembers, it's been freakishly warm.
Yeah.
Maybe they don't need a full 2 months or 3 months. Maybe you can just do January and warm them up, and they, I still think they'd breed regardless. Like corns, especially what we have here, I feel like it's probably close to like what you have with your Hondurans, where you really don't have to do much of anything.
Yeah.
To make something happen. But
and I think food cycling would play quite the role in them as well. Like you said, feeding more minimal versus, and that's the thing. I think if more people followed a natural cycle of, wintered, very minimal feeding, springtime, hit them with a bunch of little stuff, summer, go to regular, fall, start to cut it back, winter, cut them off, I think if more people followed that progression of a natural food cycle, I think that would play a much bigger role than we realize. Because I swear by food cycling with carpets. I've bred carpets. I didn't change temperatures at all, other than natural drops in a room during the winter. But I cut them off. I cut them off of food during the winter. So it's,
Have you all ever been on Bob Applegate's website?
Nah.
If I have, it's been a really long time.
as long as he's still got it up, it's definitely worth looking at to see, it's kind of in the early days. And there's a lot of really cool information. He did a lot of testing male sperm. He figured out a way to get a cheap microscope.
Oh, okay.
And so
Yeah.
he'd look at sperm motility, and he I think he was one of the ones that played around with the brumation, different brumation conditions. But he had, at least the last time I was on there 2 or 3 years ago, he still had all that information up, and thinking about it back in, I don't remember if it was the 80s or when it was. It was a while back, and he's got these this data where he's got clutch sizes by the different species he had bred, and the first clutch versus the second and third in a year, and all this stuff. He's got a lot of pretty interesting information, and especially if it's coming from so far back. And we used to, when I was in Utah, we would go to the Anaheim show, and so I got to meet Bob several times, and it's cool to hear those stories from people that were doing it, in the early days. And I think that it's been enough time that if you spend some time on that website, there might be some things there that people 20 years ago were talking about, but it kind of comes full circle again.
Yeah. Yeah. Do you do any herping up there?
I haven't done very much. I did quite a bit when I was in Utah. I spent a summer in Arizona. That was real. I saw 55 rattlesnakes in 1 month.
Wow.
I got to one day we were on a hike. So this is in the Chiricahuas. So this is southeastern Arizona, not far from the Mexico and New Mexico borders. And we're at a fairly high elevation, right where the Chiricahua Desert Museum, or their research station there. It's not far from Bob Ashley's desert museum. I think, is it the Sonoran Desert Museum? I can't remember. But anyway, he's close by there. But this research station where we were at, we're high enough elevation that it never really got much above 85 degrees. And we'd drive 5 minutes down on the flats and be 115.
Wow.
Yeah. But yeah, that was a lot of fun. But anyway, one day we were out on a hike at this higher elevation, and we saw this black-tailed rattlesnake kind of back under a rock, and we thought, hey, that's cool. We kind of shined a light in there and checked the snake out. And then the next day we were walking through that area again, and we still saw that same animal under the rock. We're like, okay, let's hook it out. Let's get some pictures. It's a sign. We see this animal twice. We've got to get some pictures. And so we hook it out. And as we're doing that, there's some babies that haven't had their shed yet.
wow.
So it was a mom with her babies that hadn't even had their first shed yet. And I was down there on my hands and knees scooping her out of there. And I realized that after I had gotten her and 2 or 3 babies out, that there was one about 3 inches from my head that was up on the side. And it had just stayed coiled up. It hadn't moved. I didn't even see it. And I wasn't even expecting babies. But after I had scooped a few out, I kind of looked around, and there's one, right by my face.
That's crazy.
That was a really cool experience. Another really amazing thing that happened. Oh man, if I'm going to be, so Harry Green had a research project going on with blacktails, and Dave, I think it was Dave Hardy was the gentleman's name. He was a retired anesthesiologist. And so he had implanted radio trackers in, I think it was 10 or 11 blacktail rattlesnakes. And so we got to go out one day with Harry Green and Dave Hardy to do this radio telemetry. And
I did that with Eastern Diamondbacks with a
Oh,
buddy
did you?
of mine. Yes. Oh my God,
that's
it's
awesome.
incredible. Yeah.
So we're out walking, and it's pinging, and it's getting more, close together. And we kind of come up over this ridge, and I can see a dead bunny in a bush. And so we got to watch the blacktail scent trail it, find it, eat it. And Harry Green sat down immediately. And this is, a world famous viper biologist, but you could tell why he's good at his job. Like, he sat down immediately, got a stopwatch out. And he said, you guys tell me when you see things, I'm going to click this. And he timed, how long it took to scent trail, how far that was, how long it took to swallow the head. He had all this very detailed information about how that feeding went. And Dave Hardy, I think at that time, he said he'd been doing it for 7 or 8 or 9 years, and he'd never seen one actually eat. And we got to be there when that happened.
That's incredible, man.
It was fun.
Oh
So yeah,
my
I definitely
gosh.
love being in the field. I just
Mhm.
haven't done it much. And the last time I was on with you guys, I said I need to come down your way and do some herping where you're at. Those are species that I've never seen, so that would be really fun.
Yeah, man, I can get you on quite a bit of stuff out here if you come in the right time of year.
My youngest kid, I think probably the biggest thing moving across the country and having young kids, but my youngest now, he's 8.5, so here pretty soon. I think he could even come out with me, and
there you go. Yeah.
so yeah, we've been kind of close by there when we go to the beach, but not quite that far south very
Yeah.
Mhm.
often. We drove down to Daytona a couple of times, so
There
we've
you
gone
go.
through there. But yeah, I need to make that happen. So if I do reach out to you randomly, that's probably why I'm probably going to ask if I can go herping with you.
Yeah, man, I'm all for it. I tell anybody, man, if you want to come down this way, let's go. I got, obviously I never make promises of like, oh man, I'll get you on everything,
Get you a
but
9-foot
I
diamondback.
I can promise to take you places that I have found a lot of stuff. So that's
Well, that's an experienced herper, is someone that
does have
knows they
some
can't promise.
yeah. Yeah.
every year I say I'm going to get out and do and herp more, and I never do. But this year, I'm going to make a serious effort to make it happen.
This year I'm dragging you because last year I got back into because like I had to
You did a lot of cruising last year.
well I did a lot of cruising late last year. That's the thing. I didn't get to go during the springtime when it's optimal because of my life, but my life got different later on in the
Things are
year.
much easier when you're single. Your schedule clears up a lot faster. Magically.
It's funny how that worked. But yeah, no. So I didn't get out. I didn't get to do a lot of herping until later in the year. So it was pretty hot by the time I got out. But then I did a lot through the fall. But even during those hot days, man, when it was raining, I was out. I was out. And
Yeah.
there was one time I tried not to go in the middle of the rain, but there's one time it was the only thing I hadn't tried yet to really see, going during a storm. And we had a big rainstorm coming through. I was like, you know what? I'm going to hit the road. Well, I went out in the middle of the pouring rain and I saw more snakes that night than I ever did,
Interesting.
any other night while it was that hot. Springtime, it's a completely
Yeah.
different story. But during the hot months, man, when it rains, it's weird. I also found doing that though, I found out ribbon snakes love rain. I never would have known that, but the only times that I have found ribbons on my road,
I've never seen one around.
they have been in substantial numbers. I mean, I found 10 to 12 of them,
Wow.
but they were only while it was raining. And it was crazy because most of them would be on the road, like telescoping, like they'd be up, have their heads up just in the middle of the road, and the rain, it was just pouring down rain. It was the weirdest thing, but it's the only time I found them is in the pouring rain.
Interesting. I definitely wouldn't have guessed that.
Yeah, no, it was odd, but yeah, man.
I'd imagine it's hard to go from herping in Utah to Virginia.
It's definitely different. We have pretty much 0 turtles and zero salamanders in Utah.
Oh yeah.
Yeah.
And here there's truckloads of both. So
Mhm.
that's been fun to see some animals I wasn't used to seeing. There's a place that we go hiking around. There's a pond, and it's kind of a little bit higher elevation, but certainly not the kind of mountains that I was used to in Utah, but
Right.
some elevation change. And yeah, I mean, that's almost every time we see salamanders and newts, and so that's pretty fun, and at least a dozen or a couple dozen turtles.
I always geek anytime I found salamanders. I don't find them often. It has not been a very common occurrence, but I found a couple marbled salamanders.
Marbleds are cool.
In
The
my
slimies, those
yeah.
are the ones that are like touched them once, made that mistake, don't have to make it again. Just
Marbles are just
Those are
amazing though. Oh my god, they're so pretty. I remember there was one time I was working on the plantation and I was flipping over some logs doing something. There's a little marbled salamander sitting there, and I just freaked the heck out. Was geeking out about this thing. And my coworker was like, "Dude, we're working. Like, shut up." about the salamander. Do your job. Like, okay, sorry. But
Nerd.
Yeah. Huge nerd.
But yeah, in Utah, we don't get like the cover flipping. Like, I mean, flipping rocks and flipping logs, like you'll see some stuff in Utah, but it's nothing like the, Midwest and East Coast where you can flip a tin, a piece of tin and see 3 or 4 snakes under it. I never had that happen in Utah.
Yeah, I've never had much luck flipping. Like I'll go through the woods and but I've never, once I have property, because I eventually want to get property, I'm gonna lay tin out pretty much everywhere I can, but I don't have a lot of like flip boards, around areas, because here, that's why like other places confuse me with how people herp. People go out and be like, yeah, I've got flip boards here, here, here. If you do that here, you're gonna get shot.
Yeah.
Okay, because anywhere you go, if it's just land, it's probably a private plantation, and you don't want to go on those like at all. So it's literally you have to have private property of your own, or you have to have access to where somebody lets you put a bunch of tin trash around it, which is not very common because everybody around here hunts and they don't want you putting trash all over their property. So it's not a common thing.
That's why you put the border of the tin down and then you take leaves and you throw it on top. No one will notice.
Yeah.
It's supposed to be more enticing for the animals.
I really need to try and get back out onto the plantation this year because I found an incredible amount of snakes while I was working out there, and I'm pretty sure my old manager would give me access as long as the owners aren't out there hunting. So
I was gonna say, as long as it's not season.
Yeah, because the owner, it's funny, man. It's a 6,000 acre plantation over in Yamasee. It's absolutely huge, and the owners are there literally like a handful of times out of the year for like a week to 2 weeks to hunt.
Wow.
And that's it. But they have a full staff, tractors, whole 9 yards, year-round work. But they come.
Wow.
They come for deer season, turkey season, duck season, dove season, and quail. So like they'll come like 5 times a year, hunt for a week or 2, and then they're gone. And it's like this is your playground, dude. Like that's insane.
Yeah.
They have an incredible variety out there. I've found some of the prettiest canebrake rattlesnakes you'll ever see out on that.
That'd be awesome.
Yeah, it's amazing.
Are you going to Daytona this year?
I would like to. I don't know for sure yet. I'm pretty sure I'll go to Tinley in March. Daytona, I would like to. I hope we'll make that happen. I think it'll probably happen.
Yes.
I'd like to go to Tinley eventually. I don't know.
You haven't made it yet, huh?
I haven't.
No, I would love to. I want to go to October Tinley because I've heard that's kind of the bigger one.
Trip.
Yeah, and I would plan it this year, but I'm planning a trip to Arizona in September, and I don't know if I'm going to be able to go to Arizona in September and Chicago in October. So, I mean,
you going to be able to herp in September?
That's what we're going for. We're going for a 7 to 10 day trip of just straight herping. We're probably going to be camping a lot.
Which part of Arizona?
The Tucson area.
Okay, cool.
Yeah, and
I've done a little bit around there.
Some of the surrounding stuff. We're talking about going to the Patagonia mountains and stuff, and
get to see some tigers.
Yeah, man. There's so many things I want to find out there. I've never done West herping. I used to live in Texas. I was in South Texas, granted,
Doesn't count.
Yeah, I was very South Texas, so it was still South, but West for me, So obviously I've been bit by an Atrox. So I've messed with a lot of Western Diamondbacks and some patch noses out there, and I got to see some of the more West stuff, but Arizona has always been like the one far West place that I really want to herp. So we're gonna try and make it happen this year.
The other really fun thing that happened in Arizona was every night we would go out road cruising. That was the main reason we were there. And I went to go take a pee before we were going to start driving, just off in the weeds. And I see this red streak moving through the grass. So I got to see a pyro up in the mountains in Arizona. I almost peed on it.
Man, that actually, talk about going to take a pee and seeing a snake. I was at work one day, and I work outside. I inspect construction sites, and I was in a subdivision. I went out down this road to go take a leak. There, I knew there was nobody back there, and I walked into the woods. And I was just about to go, and I saw a copperhead just like sitting right in front of me, and this was the only time that I have understood why people think some snakes chase you. Okay, and this has never happened to me before. So I saw this copperhead. I was like, oh, okay, like cool. Like it was a good size. It was pretty. I was like, okay, I'm gonna take a picture. It was really hot out that day. I was surprised it was even out. But I kind of bent down a little bit and leaned in to take a picture. And as soon as I leaned in, this thing like projectiled towards me, like almost jumped. It came completely off the ground and towards me, and I took a step back, and then it darted off to the side, type deal. But it was a very, very odd, and I was like, now I get it. Like, okay, I can see how somebody who doesn't know snakes would think this thing was coming after them. Granted, he came at me, then went to the other way because he panicked, and doesn't want to come at me, but it was very, very odd. But
I'd be doing the same thing if I was about to get peed
on.
Yeah.
Probably the number one species I wanted to see in Arizona and didn't was banded rock rattlesnakes. I didn't get to see them.
I would love to see them. Rock rattlesnakes are my favorite, man. Out of all the rattlesnakes, those banded rocks.
Awesome.
Oh my god, I love them.
What was your favorite species to find in Utah?
In Utah, I didn't get to see rattlesnakes very often, and I just kind of love venomous stuff, especially rattlesnakes. And there's only one species in Utah. Well, the part of Utah I was in. So the lutosis, well, if it's still called that, I haven't followed that over the years. I know it's changed some, but the Great Basin rattlesnake. We saw tons of Pituophis and tons of Thamnophis. So, it wasn't usually very exciting to see them. But yeah, really, it probably is the rattlesnakes. I didn't really see them on the roads, but going out and herping, I definitely would find rubber boas, and that was pretty fun.
Yeah.
Especially if it was like colder temperatures, and I wasn't even necessarily going herping or camping or whatever, and it's 50 degrees and find a rubber boa. That was pretty fun.
Those things are cool, man.
Yeah, they are neat.
Yeah.
It's also
interesting.
It's also funny how like herpers from different parts of the US, you say, yeah, we found Pituophis and Thamnophis, so that weren't really that exciting. Finding a Pituophis, I would lose my mind.
Yeah.
I have only ever found one bull snake in Texas, and it was just I was walking out to a spot to go herping, and this thing was going across the parking lot as I was walking up, and I lost my mind. It was awesome. I loved it, but that is the only Pituophis I have ever herped. I'm glad I can cross that genus off my list.
Yeah.
But,
well, you'll see him in Arizona.
I hope so, man. God,
we saw a decent one when we were in Texas, but it had been hit by a car fairly soon, or before, like soonish after we had come across it. So
man, I rolled this last year. This was one of the it absolutely sucked. This is the first time this has happened to me so like perfectly. I mean, I quote unquote perfect. It sucked, but someone. I obviously road cruising, you drive pretty slow, and on this road that I hit, it's gotten a little bit more traffic recently, so that's unfortunate. But I normally kind of just pull off to the side of the road and let people pass me if I see someone coming behind me. So I did. I pulled over. I let somebody go by me, and I kept going on my road. And not 2 minutes up, I see something in the road like flopping around. I'm like, well, what the hell is that? I rode up on it, and it was Nerodia. It was a banded water snake, and this dude crushed its head, went right over it, and the thing was like flopping around. I didn't know what was going on at first. I got out, and I literally watched it die in front of me. It was right there, and I was like, "Oh my god, you have got to be kidding me, dude!" It was depressing, especially because I love Nerodia, so that made me anything like that. I hate finding dead snakes, man. That's nothing worse.
Yeah,
happens.
Yeah, doing that road cruising in Arizona, we found a lot. We got to see lots of road killed stuff, unfortunately.
Yeah.
One of the craziest things that happened was there was a little, I think it was a long-nosed snake, but it was like, I don't know, 8 inches long, not very big. And we had seen it on the road. We pulled over, and then the car behind us, we watched it
Oh.
drive directly over it. Like, I saw the tire go over that snake. Like, there's no question in my mind. I personally, this isn't somebody telling me a story. I watched the tire go over that snake. And so we go pick it up, and it did have like a little bloody spot on it. But I mean, it was moving around, seemed okay. We took it back. We had a permit to be able to take any injured or dead specimens. And
Mhm.
so anyway, we were able to take it back to the research station. And I mean, 3 or 4 days later, we fed it a lizard. It ate it. It was fine, and we let it go. It was
Wow.
no problem. I don't
Yeah.
know if that just happened to be going over like a little bump right before. I mean, I don't know how in the world that snake didn't get pulverized, but it was completely fine.
I've seen a couple to where, like, you know, you could tell it had been hit, you know, because it kind of had, like, you could tell its ribs were crushed in like a certain spot, but snake was acting relatively normal, you know, and
It's amazing. They're tough suckers.
yeah, they really are. But well, we're at the hour and a half mark.
Hour and a half.
So where can everybody find you if they've been living under a rock and don't know yet?
So the website you had up earlier, raregeneticsinc.com. That's Rare Genetics Inc. is what we are on Instagram and Facebook. Those are really the only social media that we have accounts. Not that I'm very active on there, but I think through this year that will change, and we'll have a lot more going on, especially with the ability to do so much more genomic sequencing and morph sequencing. We'll have a lot more going on. So that's the best place to find us. And then for the ball python breeding and the Honduran breeding, I have another small business. It's genetic. Oh geez, I can't even. I guess it's late enough. My brain's farting. Reptile Genetic Services is what that business is. So we're on Morph Market with that, selling mostly ball pythons, but we've got a few Hondurans on there.
Nice.
Awesome.
Cool.
Yeah.
Awesome, man. Well, we really appreciate you coming on. This was a great episode and got a lot of good information out there.
Glad to have you back.
Yeah, this was a great update, man. It sounds like you've made a lot of progress since the almost 4 years ago that we had you on, which me and Justin were talking about that today. How it's crazy. This has almost been going 4 years now.
And it's fun to see you guys still doing it. And you guys have had a lot of good shows. That's awesome.
Thank you, man.
And I took the time to look up, what public stats they are, and it looks like you've done pretty good reviews and number of reviews, and that's really cool.
Yeah, thank you, man.
It's been growing.
Yeah, we've tried our hardest.
Far surpassed our expectations.
Yeah, if you would have said 4 years ago, if you would have told me, hey, y'all are going to be doing this still in 4 years, and you're going to have thousands of listens and followers, like, wait, what? Yeah. Awesome, man. Well, thank you.
Yes, thank you very much.
Yep. Thank you. I appreciate it.
Yeah, man. Have a good night.
You too.
Bye. This episode was brought to you by BlackBoxCages.com. We forgot to plug Steve and BlackBox at the beginning of the episode. We were anxious to get into it. So please give them a follow, Facebook, Instagram, all that good stuff. And Steve Snaketuary, get some Venom hot sauce, help support Steve and his Snaketuary with relocations, rehabilitations, surrenders, public outreach, all that good stuff. I think Jeff and Kendra actually just bought the whole set.
Nice.
So I think they're going to be doing a video on that soon. So that'll be cool.
Good.
And we will see everyone Monday night at 9 p.m. for Snakes and Stogies 107, I think. 107.
Good deal.
Yeah.
All right.
Thank you.
Thanks, everybody.